Again, I want to start off with this really striking view of our planet. Now you can all see it. Because I want to bring right to the front of your minds a couple of points. As I started to say, this is a multi-decade time series from NASA's Earth-orbiting satellites, and it shows the global biosphere. This is the photosynthetic biosphere, so chlorophyll concentration in the ocean and vegetation index over the land. The first thing you can immediately see as this globe turns is how much of our planet is covered by marine ecosystems. We really do live on an ocean planet. There's a lot of land right there, but wait a second, as it turns, you'll see lots and lots of ocean. The second point is not at all obvious from this view, and that is the incredible fact that these striking dynamics that we see in chlorophyll in the ocean arise from the effects of billions and billions and billions of single-celled organisms. The marine photosynthetic biosphere is almost exclusively microscopic. You see on the edge of this slide here, I've put a few examples of what these photosynthetic plankton, phytoplankton look like. These are micrometer scale images that were taken in the ocean with instruments that I'm going to tell you about tonight. The natural microscopic world is something I know many people rarely think about, so I want to emphasize that these tiny plankton matter a whole lot. They're the base of marine food webs, they contribute roughly half of global primary production, and they have major impacts on Earth's carbon cycle, nutrient cycles, they help to regulate the climate. Today, I'm going to talk a lot about plankton in the waters off the northeast US, this is an area that's warming faster than many other parts of the ocean. A lot of people, you all included, live along this coastline, and we all depend upon and strongly influence the ocean system. We interact with threatened species. We have a very long history of valuable wild catch fisheries, and all too common things like harmful algal blooms that can threaten our own food safety. All of these facets of the ecosystem depend on production by these plankton. As visual creatures on land, we're intuitively familiar with terrestrial ecosystems where the diversity of dominant plant types sets the stage for completely different animal communities, different food webs, different levels of productivity. And I want to tell you tonight that the exact same thing happens in the ocean, where in fact there's even more diversity occurring in the photosynthetic organisms. We just have to work a lot harder to observe it. So I want to give you a quick view of some of this diversity. This is an electron micrograph of a tiny picoplankton. These cyanobacteria about a micron in diameter, and they're very widespread and abundant in the ocean. And now you see, we're zooming out, you see them in scale with some example nanoplankton of a variety of different types, then some single-celled siliceous diatoms, and then eventually zooming out some relatively short chains of these diatoms. And if you have a keen eye, you might see the original Synechococcus cell at the end of that red arrow, but this might be the only projector ever where there's still a pixel that shows that cell. Everywhere else I've shown this, you just can't see it anymore. So my point is the diversity and size is enormous, but at this microscopic scale. They're also extremely diverse in shape and, of course, in their evolutionary history, their genetic and biochemical and physiological potential, the niches that they occupy, and the functional roles that they play in things like food webs. One of our prevailing paradigms in studying ocean ecosystems is this idea of large-celled phytoplankton, like those diatoms, supporting short food chains that are really efficient at exporting production to higher trophic levels like fish, while those tiny picoplankton are part of a much wider web of interactions that we often call the microbial loop, and is thought to be largely recycling in terms of nutrients. So the point here is that this is very simplified, but the different types of organisms at this microscopic scale matter a whole lot. So what are we going to do to tackle this big challenge of studying these microscopic organisms? I'm here to tell you that I think one very powerful way that we can meet that challenge is to rapidly measure lots of individual microorganisms in the ocean, and to do that in a sustained way. And my colleagues and I have been pushing forward on this front with the flow cytobot series of instruments. The initial automated submersible flow cytometer we developed was optimized for very high sensitivity to measure those small cells like Synechococcus. And then the sister instrument, developed a little bit later, imaging flow cytobot, or IFCB for short, which integrates video imaging and is optimized for microplankton, the larger organisms like those diatom chains. This is essentially a robotic flow-through microscope. For those of you interested in a little bit more detail, flow cytometry is a technique where seawater is pumped in a stream that's so thin that microscopic particles are going single file through a focused light source, that it's usually a laser beam. And there we make highly sensitive measurements of how much light is scattered and fluoresced by each particle. And it's really ideal for characterizing those small cells. Some of you working in biomedicine may know this technique. It's used extensively to measure cells in that field. But the optical signals that we get from conventional flow cytometry are really difficult to interpret when we have large cells with complex shapes. So what my colleagues and I have done is advanced a solution that combines the flow cytometry with video imaging. So just downstream of the laser beam, after they cross the beam, the particles are exposed to a short, bright flash right when they're in front of a microscope objective, and then the exposed frame from a digital camera is stored, and the result are these kinds of very high-resolution images of each particle. These images have about 1 micron resolution, so we can identify organisms to genus or species in many cases. Because we have very precise flow control and we're using the laser signals to trigger imaging, this approach maximizes rapid capture of lots of images in good focus. This is an example replay of a few drops of a real seawater sample that we analyze this way. The black region is the size of the whole camera field, and each picture you see is a separate trigger and the associated image as the particles come along in the flow. This is essentially raw data, so you can see that the image quality is pretty consistent. I've just replayed it a little bit faster than the real acquisition, which is often a few per second. And I've overlaid the images for a split second so you can see them. And in actuality, they're coming along separately, not overlapping like that. Okay, so you may know that in environmental microbiology, flow cytometry and microscopy are very important widespread techniques. They've been used for a long time. But in their labor-intensive and lab-based mode, we really struggle with chronic undersampling of the huge and really dynamic ocean I've been talking about. So the innovation here is that we've translated these approaches we're familiar with in the lab into modes that work automatically underwater. So with these robotic instruments, we can have extended unattended deployments in the ocean, and we can capture unprecedented spatial and temporal scales. These technologies are a great match for ocean observatory infrastructure, and my team has been deploying them at the Martha's Vineyard Coastal Observatory since the 2000s. This site is on the inner shelf near shore off the coast of Massachusetts. And it's a cabled facility, so we have connection to shore for power and two-way communications. We basically have a power outlet and internet connection underwater. This is a really great place to explore how plankton respond to their environment, how communities are changing, because there are very big seasonal signals. This is the water temperature record from this site, and you can see the seasonal amplitude is 20 degrees or more. And there's also interesting year-to-year variations, like whether it's a cold or warm winter, and all of this is superimposed on a long-term warming trend. So along with this strong environmental variability, we now know from our in situ observations that there's a lot of diversity in the size and shape and taxonomy of the plankton here. These are a few examples of the images we've collected to give you a sense of this diversity. As I mentioned, these have high resolution, so we can identify many of these to species. I'm super excited to tell you that that collection is a very, very small selection of the images we've collected. We've been doing this work at this site since 2006, where we've now automatically sampled the ocean with this approach over 300,000 times and generated over 1.3 billion images. So this is truly, for us, a big data problem that's prompted us to invest a lot in data and information systems. And I'm pleased to tell you that if you'd like, you can access and use all of these images and the associated data products from our public dashboard and web service system. Okay, so with these kinds of data, we're working to study the occurrence patterns of dozens of species in the marine system. But today I'm just going to focus in on 3 to tell you a little bit more about. I'll briefly describe the patterns of occurrence for that tiny picoplankton Synechococcus. It's by far the dominant of the smallest phytoplankton, and I'll contrast it with Guinardia delicatula, which is the dominant microplanktonic diatom in this system. So these are both important and very successful species in this system, but at the opposite ends of the size spectrum, and we believe have different roles in the food web. And then I'll also talk a little bit about Hemialis, which is another chain-forming diatom. To the uninitiated, it might look fairly similar to Guinardia, but it contrasts extremely. It's exceedingly rare. Guinardia is the dominant, and Hemialis, we really weren't paying any attention to it in this system because it's so rare until it shocked us about 15 years in, and I'll show you why. I'm going to use these images I show you here just kind of as icons to guide you about which species I'm talking about at different points in the talk. But I want to be super clear that that electron micrograph of Synechococcus, that was measured a long time ago in the lab. We can't measure that in the ocean with our technologies. We're only measuring the larger diatoms. We measure Synechococcus with the laser. Okay, so first up, there's our small cell in this system, which Synechococcus, which we affectionately call SYN for short. We started these deployments in 2003, so it's really exciting to be here in 2023 and share the first 20 years of observations of this organism. This is ongoing work, and we've been studying it extensively. I just want you to notice here right away a couple of things. That the main thing is that there's this striking seasonal bloom that happens each spring like clockwork in this organism. These cells are extremely abundant in summertime. More than 100,000 in a milliliter of water is common, and they drop by orders of magnitude in the winter. The average annual cycle of biomass, which you see here in red for these tiny picoplankton, it emphasizes that they really dominate in the summertime, and this is presumably linked to a very active microbial loop because these are the tiny cells at providing the bottom of the food web for that component. Now, this contrasts very starkly with what we find for that dominant diatom species, Guinardia delicatula. It has a completely out-of-phase annual pattern, and with their biggest blooms occurring typically in the coldest winter months. So for the diatom, that data I just showed you, that average annual pattern comes from a record that's almost as long. We started doing these observations with the imaging instrument in 2006. It was just developed a couple of years later. For this species, again, you see there's distinct bloom events, but they're much less regular from year to year than the small cells. The biggest blooms are in winter, but not every winter. So there's a lot of interesting questions about this record that I'll talk a little bit more about in a minute. But first, I want to just pause my story arc for just a moment to emphasize how incredible this type of species-specific, very high-resolution time series is for us as ecologists. We're just like, blown away by this kind of information. Without this kind of observing approach, we would have only sporadic, very time-consuming manual microscopy, and there's just no practical way that we could capture this range of timescales from sub-daily to decadal. So it really is the combination of a cabled observatory and the instrument technology I'm talking about that makes this kind of observation possible. Equally important, I have to say, is that here we need artificial intelligence to make sense now of these huge data sets, since we don't just have a few microscopy samples. This is another view of that same time series of Guinardia delicatula, but now I'm showing it in scale with the concentration of everything that we imaged with IFCB in this record. The diatom is a thin blue line that you might be able to see along the baseline there. So my point here is that in this over 1.3 billion image record, this whole data set is over 1 billion images, way under 1% of those images correspond to this species. So it's really critical that we're able to use, now we're using supervised deep machine learning to make this huge data set scientifically useful. And obviously we can't do that by hand because there's just too many images. So we've developed automated image analysis that lets us detect targets in the images and estimate their geometric features, including each one's contribution to biomass. And at present, we've built a 155 class convolutional neural network that's performing with very high levels of classification accuracy. Obviously, I don't expect you to be reading all those labels along the axis there. Those are the many species and genera that we're able to separate with this classifier. The precision metric, the accuracy metric I'm showing here is the harmonic mean of precision and recall. Okay, so now when we zoom back in on the diatom that we're interested in, you can see that blue line I'm showing here corresponds to the machine learning-based prediction with superimposed on top that set of red points that those show intermittent verification by human experts. Every 2 weeks, we manually inspect a couple of hours worth of images and have verification that the AI result is giving us fidelity. So we're using the machine learning for the full data set, but we're checking all along the way to make sure that the performance is appropriate for our scientific analyses. Okay, so let's get back to comparing these two plankton types. And I want to raise to the front of your minds this question: why does this diatom bloom in the winter instead of when it's warm and sunny in the summertime? And what I'm going to show you next is how we're using the highly resolved observations that we're making to not only quantify these kinds of patterns, but also help us understand what's causing them. And in this case, the high-resolution images were really important. After quite a few years of collecting this kind of data, we noticed something really important going on at the microscopic scale with this diatom, and that it was interacting with other microbes, in particular, a nanoflagellate that turns out to be a very lethal parasite of this diatom. This was not something we expected. We weren't looking for it, but it became apparent in the extensive work we were doing to manually annotate images to build our machine learning algorithms. So I'm going to show you here what this parasite life cycle looks like and how it appears in the images. So here, towards the center of the slide, you see a schematic of that life cycle stage of the parasite interacting with the host, and then on the outside is an example zoom in on an actual cell image we collected in the ocean that shows the same stage. So these parasites are tiny nanoflagellates. They're able to land on the outside of the host, and then they can squeeze between the frustral pieces and get inside the host, at which point the parasite enters a feeding stage and it consumes the entire host protoplasm. And then it rapidly begins to reproduce inside the empty frustule, producing 2, then 4, then 8, sometimes even more offspring, reportedly depending how big the original meal was. And then after they divide, they break out and disperse, but not before they leave behind this telltale trail of poop inside their poor dead host. So we can sort of see the history of demise even when the hosts are dead. And then, of course, the offspring either move down, the parasite offspring either swim down the chain and find another host to infect in the chain, or they swim through the water column and find another host to infect. I want to say that we didn't discover this for the first time. The biological detail of this particular host-parasite interaction was first described in the 1990s from some manual microscopy in the North Sea. But no one appreciated the importance of this process in the ocean and in our system until we started to look at it more carefully based on this quantitative image record. So I mentioned this was multiple years in before we figured out this was going on. But the really cool thing about the digital image archive is we could go back to the very beginning and reanalyze in a quantitative way. When we first did the parasite work, we were doing that, just determining whether they were in stages of infection based on manual inspection of a small subset of images. But fast forward to today, our deep machine learning models are now advanced enough that we can automatically classify healthy Guanardi delicatula separately from those infected by by the parasites. And I just highlighted on this is that same performance metric curve that I showed you earlier for our deep learning algorithm, and I highlighted the two classes that are associated with the healthy and infected host. These have 95 and 93% F1 scores respectively. So this is working well enough for us to do studies like this. So now, as I mentioned, the power of this digital image archive is that we can go back and reanalyze the entire record with this new algorithm. And from this, we can show that parasite infection of this host is not just an anecdote, but rather it's an important and recurrent process. And in our updated thinking now, we believe this is a critical source of mortality for this important diatom. What you're seeing here is the same host concentration time series that I showed you earlier in blue, but now it's just color-coded so that the times showing high infection prevalence are the dark colors, or the pink colors here. You can see from the color bar that it can often be 10 or 20% of the population which is infected by the parasite during outbreaks, which is really high for this fast-acting parasite. It can kill the host in a few hours or a day. So we now think, as I mentioned, that this is a really important source of mortality for this diatom. I want to mention briefly that in our work to understand what controls these lethal epidemics is we started in the recent decades to complement the image record with high-throughput DNA sequencing. And among other things, this is allowing us to confirm that the parasite is actually present in the water almost all the time, even when we can't detect it infecting the host. So the host appears to be resistant to infection, or the parasite is unable to infect the host at certain times. I'll say a little bit more about that in a few minutes. Okay, so I am a huge fan of time series like this in natural systems. The power of this approach is pretty exciting for us as marine ecologists. We're learning things with this approach that I don't think we could with any other way. But at the same time, we always are very mindful that this kind of result from one location, we have to wonder, is this a very localized process, or is it much more broadly important in this big dynamic ocean I was talking about at the beginning? So I'm happy to tell you that we're trying to tackle that head on as well. As part of our long-term ecological research, we've been able to partner with NOAA's Northeast Fisheries Science Center, part of the National Marine Fisheries Service. And we've added this kind of automated plankton imaging for the last 10 years to their seasonal ecosystem monitoring surveys that extend all the way from North Carolina up to the Canadian border. This is part of NOAA's mandated responsibility for managing the living marine resources, including fisheries in this region. So we're able to strongly leverage that federal mandate and get these additional science observations done on these cruises. Each dot on this map is a location where an automated IFCB sample was collected over the last decade. Each one of those samples has thousands of plankton images. So we've done 28 of these cruises over a decade, different seasons, and collected tens of millions of images. And with these results now and the same automated classifier I was talking about earlier, we can now do things like this, where we map the distributions of these organisms, both the healthy and infected hosts, across the whole region. I'm just showing you here this multi-year composites. We can do this seasonally as well. On the left, you see that this diatom I told you was really important in the near shore, blooms across the whole region. In fact, when we pool across years and space, it emerges as an important, the dominant diatom in the whole system. So it's not important just near shore, but across the whole region. And likewise, parasite infection you see on the right is detected throughout the region. So we can now use these kinds of results to quantitatively confirm that this is a widely important process in this ecosystem. And I'll just say again to emphasize that it's really exciting for us that the things we're learning at the Coastal Observatory, where we have these decades-long time series, are helping us understand a much broader spatial domain and important processes in the ocean. I want to extend this actually to a really striking pattern that we've found with temperature. So I'm coming back now to why do these diatoms bloom in the winter. This plot shows how parasite infection rate that was observed each day. So the parasite infection rate is on the y-axis, and it's showing how it varies with water temperature. And the points are color-coded according to host abundance on that particular day. So the dark colors are when there's a lot of host in the water. And at MVCO, you can see we find very high rates of infection much of the time, but those high rates of infection do not occur in waters colder than about 4 degrees C. These are exactly the time when you see the darkest points of the host. So we now think that these cold waters are providing a kind of mortality refuge from the parasites. For whatever reason, we still don't quite understand the biology, but this is actually very common in parasite systems, that there's a temperature threshold. And it appears that the diatoms are blooming, increasing in biomass in the winter because of lower mortality, not necessarily because it's the best time for them to grow. And this exact same pattern emerges when we look at the broad scale spatial data with the newer data sets. So very low or undetectable infection when the waters are very cold. So again, the diatoms are blooming in the winter, we believe, not because they can grow faster, but because they're dying much slower than they are at other times of year. I want to just end this little story about the diatom parasite by coming back to the food web paradigms I introduced at the beginning and the functional roles of diverse kinds of plankton. Remember, I told you this idea that diatoms support short food chains that efficiently lead to fish and birds and mammals. But the results I've just been showing you now question the importance of this classic short food web, and they instead highlight that a lot of diatom biomass in this system can be directed into the production of these tiny parasites. They're small and they're much more likely to be more connected to the microbial loop, and the classic paradigm of the short food webs doesn't seem supported by what we're finding. Okay, so I just want to emphasize again that these kinds of insights that we're getting are now possible because of the quantitative observations we have that resolve both taxa and details of interactions among taxa at the microscopic scale, with high frequency in space and time and also long extent. And with this, we're able to start updating our conceptual views and doing a much better job of understanding patterns that are emerging across seasons and years. So circling back to our overview, just to restate, we found that those cold winter temperatures are suppressing death by parasites in the diatom and allowing them to flourish at that time of the year. You might remember back from the beginning that Guarnardi delicacia is one of many diatom species in this system, and I told you that I would contrast it with the story of Hemialis, this other diatom that looks very similar in its gross morphology. The first point of contrast I want to make with this plot right here is just to emphasize what I said earlier, that at the Coastal Observatory, where we've been working for 2 decades, Hemialis is extremely rare. So this pink line here along the baseline shows the average annual biomass pattern for that species. So not surprisingly, based on the Coastal Observatory, we were not paying any attention to this species. It was, occasionally observed, but just not on our radar in terms of important in the system. And then we started detailed process studies as part of our long-term research that extend all the way across the shelf. And now 4 times a year, once in each season, I'm leading small teams. We go out in ship-based research, and we use a wide range of observing approaches to characterize both the environment and the organisms, how fast they're growing and dying, and how they're interacting with each other across this whole system. On these cruises, of course, we set up IFCB so it can sample automatically the entire time that the ship is underway. So we now, since 2017, we've collected, again, tens of millions of images that extend from the nearshore observatory near the coast to all the way out to the upper continental slope, where the ocean water is about 2,000 meters deep. So it's a very large region, and we're analyzing it in the same techniques that we're using at the Coastal Observatory. And this is what we found in August of 2019. We were, I think it's an understatement to say we were just blown away by what we found. These are some of the images in one sample of the ocean that was 3.9 milliliters. And I don't know if you recognize them there, but those are Hemialis. This was the Hemialis jackpot. And this one, few spoonfuls of seawater had way more Hemialis in it than we had seen manually annotating over 6 million images across a decade at the Coastal Observatory. We'd literally seen about 40 before this. And they were everywhere except the inner shelf and the upper slope waters. And I just remind you that this is really surprising because it was summertime. This is the time when we expect to see the small cells dominating, and the diatoms usually show up in the winter. So what's going on here? Hemialis it's supposed to be rare, and diatoms are supposed to bloom in the winter, not the summer. And once again, I'm going to use the detailed image record to help us get some answers, why we were so surprised. This bloom, as I mentioned, it was widespread. You see the distribution of biomass from the near shore to the offshore in that upper panel, and it was totally dominating. There were lots of healthy cells. You could actually see the cells were actively dividing, so they were growing fast. When we look more closely at the images, we found that this diatom cell had microbial partners, just like Guinardia, but instead of lethal parasites, their microbes are nitrogen-fixing symbionts. These small cyanobacteria, they live inside the diatom host, and they can help provide this bloom with a completely different nutrient source than other diatoms that we typically see here. So this nitrogen fixing, they're able to get nitrogen from the atmosphere, just like the microbes that live in the roots of legumes on land. So this is a trick that diatoms like Guinardia delicatula can't play because they don't have this symbiotic partner. The conditions where this bloom occurred were extremely warm. It was kind of a mini heat wave in the ocean at this time, and the ocean was highly stratified, very lots of density layers on light, warm water sitting on top of colder, denser water, and the bloom was occurring in this very shallow, warm water in close proximity to the atmosphere where, as you may know, there's like basically an infinite source of nitrogen gas. So they were able to outcompete all other organisms at this time because of this microbial interaction. So to sort of summarize all of this together, we're now thinking about the interplay of temperature and nutrients and the ways that it's regulating community structure and important food web aspects in this system. The growth season for Synechococcus, we're watching it expand as there are warmer spring and fall conditions. Basically, summer's getting longer, so we're expecting this organism to continue to flourish and to stimulate a strong microbial loop in the summertime in this system. As we've seen, the biggest Guinardia blooms in the past have happened in the coldest winter conditions. And these, I didn't talk about this too much, but they likely depend on nitrate coming up as a nitrogen source coming up from deep waters. And this contrasts starkly with the Hemialis anomalous bloom that we saw, which it was favored in really warm summer conditions when they can outcompete other phytoplankton that are nitrogen limited because they don't have the symbiotic ability to fix nitrogen from the atmosphere. Guinardia delicatula is currently really important in this system, but it may be selected against as conditions continue to warm. The cold winter periods are getting shorter and less frequent. Sometimes they don't occur at all, and we're seeing failures of those winter blooms when the parasites overwinter. Hemialis, on the other hand, we're going to be watching as we go forward to see if this kind of anomalous bloom may become more recurrent as summer marine heat waves continue to sweep through this ecosystem. So in these ways, the microbial patterns and interactions that we're uncovering are leading us directly to new conceptual views of marine food webs and the roles that different organisms are playing, and also hypotheses about climate impacts that we'll be able to evaluate going forward with our long-term ecological research. I want to just be sure that I'm not leaving you with the idea that we have this nice, tidy picture of how microbial interactions function in this system. And I want to do this, I just want to share some additional, for me, it's like eye candy of these IFCB images that emphasize that we've just begun to scratch the surface of these kinds of interactions. Our imaging records are revealing that there are a wide array of parasitic and other kinds of interactions at the microscopic scale, and many taxa are involved in these. Some of them appear to be quite host-specific, and they seem to occur in outbreaks or epidemic kind of interactions. And as I said, many species are involved, and we're going to need ongoing quantitative studies to learn more about these critical processes and interactions. I also want to note that I've talked a lot tonight mostly about photosynthetic plankton, but we can also use the same imaging approaches to study microscopic consumers. So these examples here are important players in the microbial loop. These ciliated protozoa, they're single-celled organisms, and the imaging approach is very good at characterizing them. They tend to be quite fragile and hard to sample with other approaches. So I think there's a big prospect to learn about their role in the ecosystems with this approach. Based on past knowledge and our paradigms, we typically expect that these ciliated single-celled organisms are consuming tiny picoplankton and small nanoplankton. They're just sort of flitting around in the ocean and sweeping up the little particles that they can consume. But the high-resolution images at MVCO are also emphasizing that some of these small grazers are really creative in finding food sources, and they actually can do things like attack diatoms that are much larger than they are. In this example, you see some little protozoa all around the outsides of the diatoms that are using feeding tubes. They basically insert a straw into the diatom, it puts some enzymes in there or acidify the inside of the diatom and suck out the goods. Others use these external sacs, a feeding veil that they can excrete digestive material into and then suck up the goods from a much larger food item. And then some of them have have soft bodies that they can actually extend around large things like the snake that ate the rat kind of thing, and engulf much larger things than they are. So you can see our basic idea as ecologists, we always think of the big things eat littler things, but even that's probably not such a great bet in this system. And we need to learn more about these kinds of interactions. To be sure, we're not the first to document these, but the imaging approach will allow us to be quantitative about how often this is occurring and begin to try to understand the impact it might be having in the ecosystem. Last point about this is that just like those deadly nanoparasites I talked about, it adds more questions to this view we have about diatoms being principally consumed by large zooplankton like crustaceans. These smaller protozoa that are eating big diatoms, just like the nano-sized parasites, are exporting diatom biomass into the microbial loop and adding steps to the food web and changing the balance of energy flow. There seem to be numerous mechanisms that we need to learn more about that are changing the structure of the food web. Okay, I have been kind of overwhelming you with a lot of basic research that we're doing with these technologies and the kinds of new knowledge that we're gleaning from high-resolution observing strategies. And I hope you understand I focused a lot on the data sets that my own team has collected. But I want to take a few minutes because to tell you that I think it's very exciting now that these kinds of time series and spatial observations that I've been talking about are no longer unique. So we've been investing in technology transition that's promoted a broader use of the technologies, And I will also just take a minute to highlight an example of how these technologies have been applied in a case that's got quite immediate societal relevance. So when my colleagues and I first put IFCB, the prototype, in the seawater test well in Woods Hole in 2005, I promise you we were not looking ahead to today. We were just thinking it wasn't going to work the first time we put it in the water, and it actually did. So now there are over 100 IFCBs being used around the world. It's definitely been interesting and rewarding to follow this path and watch the evolving uses. The technology has now been licensed to a small company, and they produce it for users around the world. And we continue to work with them to keep the technology fresh and to support the growing user community. I'm sure it's obvious to you from this talk that our original motivation for developing this technology was our basic research of plankton ecosystems. But it quite rapidly took on this other life that, as I mentioned, has direct societal relevance. And, now, instead of there being just this one Martha's Vineyard Coastal Observatory site that we're studying off Massachusetts, today there are IFCB time series locations all along the US coastline. These are being operated by a community of users. And these sites are principally being used to detect and understand the progression of what we refer to as harmful algal blooms, so phytoplankton that can produce toxins or other effects that impact the ecosystem and can also affect human health. Here now I just overlaid on top of the time series locations the occurrence patterns for various shellfish poisoning outbreaks that are associated with toxins that these phytoplankton can produce and that accumulate in the food web. And I'm sure you've heard in the news of red tides and other impacts that gets a lot of attention. And in fact, consuming seafood, humans consuming seafood that has accumulated these toxins from the plankton can land you in the hospital or even be lethal. So this is very, very serious stuff. The growing group of IFCB users are deploying their instruments in really creative ways at local piers, at other nearshore sites, on moorings offshore in the ocean, on floating barges, and there is even a user using an autonomous surface boat, an unmanned surface boat with an IFCB on it. And the IFCBs, they're imaging particular taxa that are known to produce these toxins and watching to see where and when blooms might be occurring, so that information can help to monitor for ecosystem impacts and food safety. I just want to mention that one of the really powerful aspects of this approach is that because the IFCB images aren't just measuring the toxic species, they're measuring everything in the water. We're not just monitoring for the toxic species, but we're also coupling it with research to understand how the communities are changing and what may be causing the blooms and how they're responding to environmental conditions in a more holistic way. It's also very exciting to say that these kinds of observations are being made across the globe now at an accelerating pace. The time series locations are expanding, and shipboard observations, like I talked about tonight, are also expanding around the globe. These include some sustained approaches, like in Europe, they're putting IFCBs on regular ferry crossings to monitor for ecosystem change in the ocean. And by my conservative tally, working with the community to bring together these publicly available data sets, we now have used this approach to sample the ocean over 1.2 million times and generated billions of plankton images. And there's really an exciting culture of data and algorithm sharing among this growing community of users, and I think it's going to be super exciting, next coming decades as these data sets continue to grow and we learn unexpected things from them. First, I've stood up here and told you about a whole bunch of research that is intensely collaborative, and I want to just close by expressing how grateful I am that I get to spend my days surrounded by these smart people. They take care of each other as coworkers and friends and sustain the observations very passionately. It's hard work to to produce time series in the ocean, and it takes a lot of dedication. And we have a lot of fun synthesizing our understanding and growing knowledge together. And I want to extend a really big thank you to all of them. So I've tried to show you tonight how imaging approaches give us the tools that we need to meet spatial and temporal scale challenges in the ocean at the same time that we get critical taxonomic resolution at the microscopic scale. This is just, I can't overstate how important I think that is to help us understand patterns and processes of change in this important part of our planet, how they're linked to environmental drivers, the way that microscopic organisms are interacting, and what the biogeochemical impacts of those interactions may be. And taking together, I think it's really exciting that this kind of approach is leading us not only to new insights and mechanistic understanding, but it's bringing up new hypotheses and helping us to be poised to understand how our ocean system is changing in response to ongoing climate change. And with that, I want to say I'd be happy to talk with you about anything I presented or answer questions if there's time. And I want to thank you all very much for your kind attention. Please.
So 2 questions. You showed that example there where you have a little guy inserting a feeding tube into a diatom. And you said it sucks out the contents. So the question is, how does it physically produce the suction?
I have no idea. Does anybody else know about that?
It's not like a muscle that creates a bigger bladder, reduce the pressure.
There's no muscle. These are all single-celled
right,
organisms,
yeah, exactly.
so there's no muscles involved.
Okay, so my second question was, so all of this refers to basically unicellular life. Is there some kind of a knowledge base like this for viruses in the ocean, and maybe even for things like, I don't know, prions or something like that?
Yeah, so I don't know about prions. For sure, viruses are important in the ocean, and they're not something my research group is working on directly, but they're equally important to parasitism and other kinds of interactions. Viruses are well documented to change food webs, to produce different pathways of carbon and energy flow, and to impact species composition. They have very big impacts in marine ecosystems, and they're an active area of study. It's not my area of research, but it's definitely very important. I didn't mention that when I showed you the slide of other interactions, that wow, we have so much left to do. I think a bunch of those organisms that you could see on the outside of diatoms are actually fungi. So they're very poorly studied in marine systems, but it seems like fungi are also really important and underappreciated, need more attention. The more we look, the more we find at the microscopic scale in terms of diversity, and the gene pool is vast, for sure. Oh dear. Okay, I think that we'll go back here, and then I'll come here, and then I'll go back to there. Go ahead, please.
Or is anybody applying methods to computationally infer, like trophic networks, or building dynamical models that could predict future time points in this very high-dimensional compositional space? And also, like, are the data open for access yet?
Okay, so there's quite a bit there. We're definitely just scratching the surface of trying to use those types of modeling approaches. I had a couple of interactions with students who have been working on things like topic models to help us try to understand community assembly and how the communities are changing through time. More complex dynamical models we haven't explored in detail at the level of species and genera. It's part of the long-term ecological research that I'm helping to lead in this study site. We have collaborators who use numerical models that are much more aggregated, large and small plankton at different trophic levels, and that are those models are coupled. They're food web models that are coupled to dynamic models of the ocean physics and allow us to look at the whole system holistically, but those don't go down to the level of resolving individual species and interactions of a species with its specific parasite, for example. So there's a lot of potential to be tapped there, and happy to hear ideas you might have about how to be productive in that direction. So I think you asked if the data are open access. So a whole bunch of the data all of the raw data that we produce and the sort of first and second level products that we're producing are easily and readily accessible. The kind of final products where I've interpreted the output of a classifier and made a time series and painstakingly kind of checked that I trust it, those are, the underlying data is available, but you'd have to do your own interpretation to generate the time series right now. We're working actively to try to be better about sharing those higher level products that take a lot of investment and care to interpret. We can talk more about it after. I'm a very avid data sharer and would love to share products with you if you have ideas about how to use them. Did you have another question, or did I get them all? Maybe, okay, thanks. Right here, please.
That was my favorite talk that I've ever heard on oceanography across the spectrum.
only talk he's ever heard on oceanography, right?
Actually, my teachers were Chandler Fulton and Philip Klein at Woods Hole when I was at Brandeis and Harvard Medical School. So I'm very thankful. So you refreshed me again on cyanobacteria are basically chloroplasts, and the chloroplasts are basically pre-mitochondria. So there is a way to get macro picture information. But then as I started to listen to you in more detail and study, I realized that we are very far from an ocean model like we have with Drosophila at Columbia or C. elegans at Rockefeller. There must be a way to isolate a few square miles of ocean, use your AI, your artificial intelligence data, and hold things or hold smaller aquariums that you can study because you have no macro. I keep looking for it. The macro data, everyone guesses the ocean's circulating in 1,000 years based on currents, but I don't see any diatomic or radio-labeled plankton to help us tell whether or not the ocean. And the ocean's much sicker right now than people are willing to admit. I mean, you have so much plastic that it's ridiculous, and you have so much death and problems. So I don't feel that you're going to get to that stage of what we do in medicine, which is a whole body scan and repair. So how are you going to get to a model of the entire ocean from this?
Okay, well that's a loaded question. So I don't have the hubris to think we're going to cure the ocean with a whole body model. So I'm a basic researcher and studying things that I'm fascinated in about how the ocean works. There was a lot to unpack in your question. Let's see where to begin. I think for sure humans have impacted the ocean in ways that many of which are very deleterious, But what I wanted to say about the deleterious impacts on the ocean, I know there's a lot of sensationalism around, the ocean is dying. And I don't want to minimize the bad impacts that are happening and very evident at higher trophic levels. But some of the microbes are extremely resilient. They grow fast. There's a lot of diversity. The microbial ocean is not dying. It's changing, but it's not going to die on us. We're going to be gone before the microbes in the ocean are. And they were here long before us, shaping the planet, and they will be here long after us. I feel pretty confident saying that. And I think we'd be better poised to learn about it than try to manipulate it more. But that might be a very, I don't know. Let's chat after. Okay, I think there was a question back here, and then I'll go back there.
I actually had two questions. One, I think you started to answer with just about overlaying the time courses that you have at your level of, at your size range with sort of larger size ranges. I was just curious if you saw, or if there's an effort to correlate the changes of different, zooplankton or whatever, going forward. The second question was just whether you have found that for climate change, for overall warming trends, whether what you see in the Northeast is now paralleling what you would have seen further south, or is there something different because it's just a different starting point?
Yeah, those are great questions. Okay, so I love your question about connecting the kinds of data sets I showed you tonight to, okay, what's happening in the rest of the food web. And part of our long-term ecological research is trying to do just that. And we're taking a couple of different approaches. I mentioned briefly the collaboration with the National Marine Fisheries Service. They have decades-long records of higher trophic levels in this region. They've been studying it since the 1970s as part of their mandate to manage the living resources. And we are collaborating directly with them to try to work on connections between change at the bottom of the food web and change at the commercially important fish and other species in the system. There's a bit of a gap in the middle of all the, the things that eat the phytoplankton that then the fish eat. And we're trying to tackle that with some new approaches I didn't have time to talk about tonight. One of them involves imaging in the ocean with a larger scale. So we're able to now, we tow a system behind the ship and move it up and down through the water column and collect 15 frames a second where we're imaging the shadow of every organism in about a 3-liter volume of water 15 times a second. And we're using that to be able to get images like these of the bigger animals. So copepods, gelatinous organisms, a whole array of consumers in the system. And we have about 3 or 4 years of that data seasonally, and we'll be continuing to collect that and trying to piece this all together. Your second question was about, climate, yeah. So sadly, I think it's fair to say that for the kinds of plankton that I'm studying, we don't have extensive enough records of different sites to evaluate that question. I think that network I showed you of sites all around the U.S. coast, a decade from now, ask me that question again, and we'll be starting to piece together some of that, those patterns of change latitudinally as warming is affecting the ocean strongly. I can say that those long records that NOAA has collected show absolutely that larger animals are changing. Their range distributions are moving, in some cases, northward to and also deep into deeper waters, and presumably in search of thermal habitats that match their needs for survival. So there's definitely patterns like that going on in the upper trophic levels. Okay, I think back there, and then I'll come to
Last question I got.
Oh, were we out of
We're going to
time?
be
Sorry.
running late after that.
Oh, all right, in the back.
So my question is, in terms of your food web structure, where do krill fit, and do they consume the phytoplankton or the protozoa? And the overall decrease in krill population has to do with the overall decrease in the microscopic organisms.
Yeah, so my food web diagrams were super simplified because I wanted to, be able to make some points that without getting too complex tonight. But krill would be kind of between the copepod that I showed eating the diatoms and the fish. And they're definitely important in these systems. So there's something that we are studying with that new imaging system I just mentioned. So they're definitely part of the picture. In the Northeast U.S. shelf, copepods really have a big effect. Krill are important in other ocean regions, like almost more important than copepods in some cases, but the copepods are really dominant in terms of the food webs in the Northeast. And one more, or you want to stop? Okay.
So I have a question about the single cell ones. The things you're studying don't seem to need land, so to speak.
Land? No.
So
land.
No. So would these be similarly distributed deep in the ocean? And then what brings the land dependence? So clearly, fish live closer to coastal areas. So at some point, there's some influence of the coast. So where does it come into this thing?
Okay, I love that question. Okay, so The photosynthetic organisms that I talked about, they don't care about land, that they do care about sunlight. So they live only in the upper layer of the ocean where enough sunlight penetrates that they can still photosynthesize. Other microorganisms like heterotrophic bacteria and some of these protozoa can live at all ocean depths. But typically there's higher abundances of them near the upper ocean because that's where the base of the food web, the photosynthetic productivity is so high. In terms of why there might be more plankton closer to land than in the middle of the ocean, I think the simple answer to that is the biggest issue is the supply of nutrients. Just like you fertilize your garden to get your vegetables to grow, these microorganisms need nitrogen and phosphorus, the diatoms need silica.
But you had something that fixes nitrogen from atmosphere and so on and so forth. So I presume there is an ecosystem that doesn't need the supply of nitrogen from land.
From land, right. So in the ocean far from land, there actually is a huge reservoir of most nutrients in the deep, dark water. So just like you have, leaves and other material degrading on the forest floor in terrestrial systems, you have the same kind of microbial degradation in the deep waters and nutrients accumulate. So anything that brings deep water up will fertilize the open ocean. So the upwelling systems tend to be high productivity areas. The nitrogen fixing, we classically thought that was important in the open ocean where you're far from nitrogen sources, other nitrogen sources, and there are definitely strong evidence that nitrogen fixing symbionts are important in open ocean areas. This idea that they might also have a big impact in coastal zones is something that hasn't really gotten much attention previously. Might be an impact of climate change, making the system more strongly stratified and making it harder to come by nutrients even near the land. With that, I think they're going to drag me out of here. So thank you all again. It was lovely to talk with you.